different. We saw that, for example, methylated lysine on position 9, so this is what is written on the chart just on the right, give rise to gene silencing. So it means heterochromatin formation, no transcription possible. Whereas on the opposite side, if we have an acetyl group that is directly cross-linked to this lysine at the same position, position 9, so it's written in the chart here, you have deposition of enzyme and of histone, and anyway, it's a situation in which the chromatin is open to transcription. So we saw so there are plenty of post-transcriptional translational modification in the histone tails. So it's very complicated, and there are some subtleties that are very difficult to understand, but we're going to stay in a quite simple situation. So from this post-translational modification that we can find in the histone tails, I showed you a little bit what was happening in two kinds of, this is the most common and the most studied post-translational modification of histone. So I talked to you about histone acetylation. So the acetylation of the lysine at position 9, for example, of one histone is done by an enzyme which is called histone acetyltransferase, and you can see the reaction. So there is an acetyl group that is linked to the amino group of this amino acid, and the consequence was the shift from a condensed chromatin to an open chromatin. The condensed chromatin was not open to transcription, and the closed one is the open one is open to transcription. So this was one example concerning the acetylation of the histone. So we saw after, and I made another slide because I understood that yesterday it was a little bit confusing for you. We talked about this second possibility of remodeling the chromatin. When I say remodeling, it means changing the state from a condensed to non-condensed, or the other way around. So what we saw just before with the acetylation of the histone, the consequence is if all the histone are acetylated in the genome, the whole genome will be completely non-condensed. But we saw also that there are some specificity, some situation in which, for example, there's somebody, in which we are in a place in the genome that is composed only with heterochromatin, but in this loci in the genome, we need to have gene that can be expressed. But because this part of the genome is compressed, is condensed, all the machinery of transcription cannot reach the DNA. So there is an alternative to global decompensation of the chromatin, which is just decompensation at the local place. Okay, and we saw that it was a kind of summary. So we talked a little bit, and I will show you a slide to explain you a little bit, because we call transcription factors proteins that are able directly or indirectly, that's the key point, to modulate transcription. In the case of the chromatin remodeling complexes, I gave you the example of the SWI-NSF complexes. We have, this is this small ball in black, a protein that we call a transcription factor, but this protein doesn't have any activity in terms of activating the RNA polymerase. What this protein does, it can recognize a specific sequence on the DNA, and it can also interact with enzymes that are able to modify the N-terminal tail of the histone. So in that case, it's a histone acetyltransferase, which means technically that this transcription factor, the little black box, will be able to recognize a specific loci in the genome, but because it is in interaction with an enzyme that is able to modify the histone tails, it will change the nucleosome, and then it's able to just to swipe the nucleosome in order to reveal the important sequence, for example, the data box, in order to start transcription. So it's a model. It has been discovered in yeast. It exists in superior mammals as well. So the idea here is using a protein that has the ability to target a specific region in the genome, and because this transcription factor is harboring histone modifying enzyme, indirectly it can change the structure of the chromatin locally, and then the consequence will be activation of transcription. So we saw that. I did the slide to do the comparison between direct activation and indirect activation. After, we saw very quickly another type of histone modification, which is the methylation. So I just want that you remember these two types, methylation and acetylation of the histone, because there are plenty of other post-translational modification, but it will be too complex and very complicated to make the synthesis. So only acetylation and methylation. So in this slide, I showed you that the methylation can occur also on amino acids that are basic, like lysines. And this lysine can be the lysine at position 9, because this position 9 can be either acetylated or methylated. So this is the simple methylating process. It is catalyzed by enzymes which are called histone methyltransferase. Okay, so what you see in this reaction, it's a very simple reaction. We've got the amino group of the lysine, and then there is a first linkage of one acetyl, then two acetyl, and at the end, the version of the lysine contains 3 methyl groups, and this is what we call methyl lysine. Okay, and the consequence on chromatin, so we saw this text yesterday, the consequence of methylation of histone is that the chromatin is going to be completely condensed. So the methylation of histone tails, this is the name of the enzyme that is catalyzing this reaction. This is a few examples, and I explain you why I show you this example of protein that have an histone methyltransferase activity. Okay, and I talked to you a little bit about this concept of writers and readers. I hope you did understand. So just as a summary, what we call the writer are typically this kind of protein, like histone acetyltransferase, both of them, because this is enzymes that are able to write marks on the histone. And these marks, they can be, in the case of histone methyltransferase, there will be the methylation of histone. So, for example, we call that there is one common feature of all this histone methyltransferase is that in terms of structure of the protein, they contain a domain that we call set. And the name set, S-E-T, is just the acronym of the first 3 methyltransferase that has been identified. So S stands for SUVA. It's something that has been found in Drosophila. I will show you exactly the example of SUVA. Then this one has been found in yeast, and this one is also in Drosophila. And the acronym SET, S-E-T, comes from the first letter of the first 3 histone methyltransferase that has been characterized. Okay, and we have the writers, and the readers. The readers, what they are? They are system, protein, complex of protein, or whatever, that are able to identify, to read the state of methylation of the lysine. So if a lysine at position 9 is methylated by a writer, for example, the HP1, which is the acronym is heterochromatin protein 1, this HP1 is able to read the state of methylation of this lysine in histone H3. So do you understand better the concept of writer protein, reader proteins? It's important that you answer me for that. Is it okay or not? It's okay. So we have two partners, one that is writing the code and the other one that is reading the code. Okay. So all this data, we keep that in mind. So this was a little bit of detail on how it works. And here we've got typically an activity of reading the state of methylation of histone H3. So I explain you. Here we've got a mature nucleosome that is composed by the 8 histone. The lines that are going out are the N-terminal tail of the histone. And so, for example, in this drawing, you see that histone H3 has been methylated here, the little red box, on lysine 9. So it has been methylated by a writer protein, okay? One of these SET protein. So it has been methylated by a writer, and HP1 is a protein that is able to read this state of methylation. And in this drawing, you see that, for example, the methylation of lysine 9 of histone H3 will allow the interaction with HP1, and the consequence of interaction with HP1 will be the formation of heterochromatin, so no gene expression. Okay, and so here in this drawing, it's more complex than what we need to know, to remember. You have the example of different readers of chromatin marks on histone H4, and you've got one here, which is a reader of the state of methylation of lysine 20 in the case of histone H4. Okay, so these are just examples. So technically, what I want you to remember is the concept of the modification of the tails of histone are done by enzyme. It's reversible process because you can, in the case of acetylation, you can add an acetyl group and you can remove it. These are two different enzymes that are doing this job. You have methylation, exactly the same process. So enzymes that are able to link a methyl group or to remove the methyl group. So reversibility as well. So the enzymes or the complex of enzymes that are adding acetyl group or methyl group, they are considered in the category of writer enzyme because they put new marks or they remove, I mean, they change the code. And then on the other side, you've got all the complex of protein that are able to read this state of either methylation or acetylation. And it's the reading of the state of acetylation or methylation that will have a consequence in the compaction or decompaction of chromatin. Is it okay for you? If you don't understand, do not hesitate, please, to ask me a question where you didn't understand. It's okay? Okay. Then, okay, so this one we didn't see it yesterday. So hold on, because I've got a problem with my. So it's a kind of summary of what we saw now. So the drawing, we have to read it either from top to bottom. I will put the pointer. Either from top to bottom or bottom up. It's up to you. So anyway, we have two situations. The one on the top, it's clearly written there. It's euchromatin. Euchromatin, it's relaxed, non-condensed. Transcription is active, is possible. Can be. It depends on transcription factor, obviously. And you see here, well, it's a kind of schematic. The histone state, we're going to focus on histone H4, which is one of the best known histone. So in euchromatin, histone H4 is mainly acetylated. So for example, the first one is this famous lysine at position 9. Okay, it can be phosphorylated, can be acetylated. Anyway, the chromatin is open, transcription is on. And here you've got the couple of the two enzymes, which are writers. They are belonging to the family of writers. So in one direction of the reaction, we've got, so this is the case, for example, for phosphatase, but we have also for acetylase. We've got enzymes that are able to put the mark or to remove the mark. So one example here is for the phosphate, for example. We didn't talk about that, but it's exactly the same concept as acetyl group. So in the case of acetyl group, the reaction catalyzed to add the acetyl group is catalyzed by histone acetyltransferase or kinase in the case of the phosphate. The opposite reaction, which is the removing of the acetyl, is catalyzed by histone deacetyl complexes, and removing of the phosphate will be done by phosphatase. So I just tell you because it's written there. So here we see that this reaction of writing the mark can be done in both ways. Either you write the mark or you remove the mark. So these are writer complexes. So all chromatin, specific chromatin marks, transcription possible. Then if you remove and or change the marks, so in this drawing, first step, we remove the acetyl marks and the phosphate, but don't take care about the phosphate. We remove the acetyl group of H4 and we add, you see, this is histone methyl transferase. So it's the enzyme that is able to link a methyl group to histone H4. Sorry about the drawing, but it is histone H4. So here again, it's a writer because you are writing a new mark on something that didn't have any mark. Okay. So first step, you remove the acetyl mark. Second step, you add the methyl mark. So the consequence is nucleosomes that are harboring methyl group on histone H4. And the histone H4, when they are methylated at position 9, as we saw before, this protein can be, this state of chromatin, this state of post-translational modification, can be read. So HP1 here is a reader of the state, can be read by different protein. And in that case, in that drawing, you see, we show the interaction between histone H4 when it is methylated that can interact with HP1. And one property very important in chromatin dynamic is that HP1 is able also to interact with itself. So according to you, when you see this kind of schematic, what can be the consequence in terms of chromatin condensation when you see this kind of model? Can you try to tell me if you have any idea? One of you want to try to explain me? We are here to discuss, don't worry, there is no...
So maybe you...
Maybe speak a little bit louder because...
Yeah, maybe they will be more condensed. And the transcription process cannot occur because the protein cannot reach the DNA.
Because it is condensed.
Yes.
But yes, so you're right. So you're completely right. So one of the, so you see here the clear link between you change the post-translational modification of the histone. And if we look at the system, the global consequence is once the histone H4 is methylated, it interacts with HP1 and HP1 interact with itself. So we are shifting from an open state of chromatin, and the fact that HP1 can interact with itself, it means everything will be compact. So this is the molecular mechanism, one of the molecular mechanism of compaction. Okay. So can you really understand the link between histone tail post-translational modification and chromatin condensation? Is it okay for you?
Yes.
Okay. So yesterday we talked about this local remodeling of chromatin, and one of you asked me a question, but because we start from the context, the concept is like this. Imagine that we are in a place of the genome where the chromatin is extremely compact, and the cell need to activate one gene which is inside this compacted chromatin. So I told you that there are some specific factors, so some transcription factor, but we're going to see that under the name transcription factor, actually there are two different categories of mechanism. So the one that is explained in this slide is how a protein that we called a transcription factor. So it means that this protein is able to activate one gene. How a transcription factor can activate a gene without directly activating the RNA polymerase, but with locally decompacting the chromatin. So I try to summarize how it works in this picture. So we have to read it from top left there, and then we go down to bottom right. So the situation at the beginning is compact chromatin. So the little dark line that you see here are the histone tails. Okay, it's a kind of schematic. Anyway, the little green balls there, they are those famous transcription factors. So what are they? They are protein, okay, that are able to recognize specific DNA sequence. They do not recognize histone modification, whatever. They recognize specific DNA sequence. How can they recognize the sequence? Because DNA, it's compacted completely. But keep in mind that the nucleosome is a kind of ball, and DNA is rounded around the nucleosome. So there are DNA sequences that are accessible to external factors. So in that case, we have one complex of protein or one single protein, it depends on the situation, that is able to recognize a specific DNA sequence, but this specific DNA sequence is contained in heterochromatin. So it means no transcription is possible at this place. And the function of this transcription factor will not be to directly activate RNA polymerase. So sorry, it's not written, but RNA polymerase complex is this big blue balls, the complex. So this transcription factor does not directly interact with RNA polymerase to start the activity. But what it does, this is what we saw when on the slide where I showed you the three SNF complexes. This transcription factor that is bound to DNA can interact directly with complexes of protein that are writers of epigenetic marks. It means that, for example, this transcription factor can interact with histone acetyltransferase, histone demethylase, anyway, interaction with writer complexes that are going to change the post-translational modification of the histone tails at this locus only. So just at the place and around the place, but not very far away from that, where the transcription factor recognizes the DNA. So the consequence of this is that the transcription factor, technically, it does only one thing. It's just find the right DNA sequence to bring at the same place enzymes that are able to modify histone tails. So those enzymes, they are going to change, for example, acetylation of histone H3 or demethylation of histone H3 and so on. So the consequence will be open chromatin, but open just at the locus where the transcription factor were able to bind. And from that, it means that now all the machinery of transcription, RNA polymerase II, all the general transcription factor, they will be able to directly bind to DNA and then start transcription. Is it okay? Is it clear, more clear than what I explained you yesterday? So you see, we have the possibility, the cell has the possibility to locally decompact chromatin in order to leave the place for all the necessary machinery to start transcription. And this is very important in terms of genome stability. It's a little bit what I was telling to you yesterday. There are big regions in the chromosomes that are only composed of heterochromatin because they contain a lot of transposon and repetitive DNA sequence. So the cell needs to keep this locus completely packed in order to avoid transposon jumping that can be completely deleterious for the genome. But this type of region, they may also contain very important genes that are necessary and compulsory for the living of the cell. So instead of completely decompacting the whole region, that can be dangerous because transposon can jump, the cell is able only to target one specific region of this condensed region just to decompact it locally and then start transcription. All right? Okay. Then, yesterday you asked me what's happening in that situation during transcription elongation. So we are keeping the same context. We are in heterochromatic context, so condensed chromatin. A gene needs to be expressed inside this heterochromatin. So the first event are just the one that we saw in the previous slide. So local decompaction at the site where the RNA polymerase needs to bind to DNA. But what we have to consider is that all the rest of the gene, the coding sequence, exons and introns, they are still in a compact form. So the transcription, the RNA polymerase need to go on transcribing the whole gene. So there is a process, and this is what is summarized in this slide. So this is the two-step C and D. The complex number C, so the drawing, so it's coming from this paper by Workman in Gene and Development. It was on yeast in Saccharomyces cerevisiae. So if you look carefully, it's not very easy to read this kind of drawing. Can you see that there are two different ways of drawing the nucleosome? Tell me just if you can see them. There are this one, there are big balls, okay, and this one, there are small balls. You see?
Yes.
Okay. So the beginning, the initiation, so pre-initiation complex. It is everything that is happening around the data box here. So the little arrow here is the transcription start. In blue, you've got the complex of RNA polymerase, okay? And here in dark blue, yellow, green, they are what we call the general transcription factor. We will see them on Friday. I will explain you over there. But anyway, in this drawing, what they want to show you, sorry, we are in the context where initially the chromatin is compact. So it is heterochromatin. We had, just before this state, we had one transcription factor that came in that loci and locally, as we just saw before, locally decompact the chromatin in order to have a pre-initiation complex, so Pol2 plus all the cofactors that are able to bind to the Tata box. So now, in that situation, the part number C, we have everything that is able to start transcription. RNA pol, general transcription factor, and this dark line is, which is written CTD, it is the C-terminal domain of RNA polymerase. It's a very important part of the RNA polymerase too, because it is the place where other factors are going to interact with polymerase in order to modulate its activity. Okay, so initial state, everything is ready to start transcription, but we have a problem. What needs to be transcribed here on the right is still in compact chromatin state. So it means that even if everything is ready to start transcription, the polymerase won't be able to read because it's completely blocked. So what's happening in order to have transcription extension is that, so look at the small drawing, so heterochromatin with the round ball, heterochromatin on the left, on the right, sorry, at the beginning. And what's happening is that when RNA pol starts, so this black line, it's written here, it's the new RNA which is synthesized by the Pol2. What's happening is that there were local decompaction. You see here, it's a nucleosome which is not compact, but polymerase can go on transcription because it is interacting with something that we call elongator. But the name elongator was given before knowing the dynamic of the chromatin. And actually, this protein that historically was called elongator is actually an histone acetyltransferase. So how does it work now? So you have the polymerase that needs to go on transcribing, so it needs to move along the DNA, but because of its C-terminal domain, that is in interaction with an histone acetyltransferase, what is going to happen? It's going just a little bit before the Pol2, it's going to transfer acetyl group to histone H3 at position 9 and transfer acetyl groups. And the consequence of acetylation of histone is decompaction. So in other words, you've got the polymerase that is moving, and just before the polymerase, it carries an histone desacetylase, acetyltransferase, sorry, in order to decompact. So actually what's happening is the process is completely dynamic. The decompaction happened just before the place of the RNA Pol2, and it goes on moving like that during transcription. Do you see what I tried to explain? Is it okay?
It's okay for me.
It's okay. And on the other side, so beginning, everything is ready to start, transcription start, and as transcription is going on, acetylation is done on the new reads, but at the back, there's some enzymes that are going to remove the acetyl group in order to recompact the chromatin that has already been read. So in other words, it's like the very beginning of this type of activation, the very beginning of activation of transcription is local decompaction. This local decompaction, you have all the pre-initiation complex that is able to bind to DNA. And the second step is elongation of transcription. And this extension step of transcription you can consider it as a kind of window that is moving with the polymerase. And this window in front, you add acetyl groups to lysine, so you decompact, and at the back, you remove the acetyl in order to recompact again. So it's a window of decompaction that is moving with the Pol2. Okay? Does it make sense for you?
Yes. So yeah. So combining, I just am not sure if I understand correctly. So combining the two figures, the one on
This
the
one?
previous slide, this one,
Mhm.
yes. So as I understand it,
Yeah.
at the beginning of this figure,
Here.
yes, the chromatin is
Compact.
heterochromatin,
Yes.
very, very compact,
Yes.
twirled and all of that. And then when a specific transcription factor recognize a DNA sequence, then it opens the chromatin from heterochromatin to euchromatin.
Exactly. This is this step. Okay.
Yes.
Okay. Then
And then
it's
yeah,
open.
at the end of this figure is the binding of the transcriptional complex.
Yes. So
Yes.
at the end of this figure, this last step, I'm sorry, because this figure is the beginning of the next one. So
Yes. Okay.
as you explained, compact, reconnection of the site, decompaction, then all the blue things here is the pre-initiation complex that we found here. So technically, this last part of this slide, you see my mouse?
Yes.
Okay, so this situation here, we have RNA Pol2 plus general transcription factor. Chromatin is open, but just at the data box. And this step, it's exactly this one. So it is, you see, compacted on the right, compacted on the left, non-compacted, and this is Pol2 plus the general transcription factor. In other words, the blue, light blue, dark, yellow, and so on, they are all this blue one.
Yes.
Okay?
Okay.
So local decompaction just at the site of interaction. And then next step that is not on the previous slide is how this complex can move, can slide.
Yes.
And the sliding is done by acetylation in front and deacetylation at the back. So it is what I was telling to you, like a window of decompacted chromatin that is moving with the pole. It's okay?
And the open part is like completely open, or like euchromatin open.
It's euchromatin. It's
Okay.
you never have. I mean, it's a little bit not true. You never have naked DNA. You have naked DNA. There is only and it never happens at the same time. The only moment in the living cell where you have completely naked DNA without any nucleosome, it's one step at the meiosis where we can have crossing over and exchanges, but it's always engaged with nucleosome.
Yes. Okay. I understand now.
But you understand this sliding window?
Yes.
Okay, so you decompact in front and you recompact at the back in order not to have everything which is decompacted.
Yes.
Okay. Perfect. So we go on a little bit. So this is a summary of all what we said now. Okay. So how? So I'm not going to spell. Sorry, I lost my mouse. So we're not going to repeat the things. So that's the summary. How transcription initiation can occur in two contexts: euchromatin, heterochromatin. So euchromatin, you know it, direct access of transcription factor to Pol2 and local remodeling, like we saw. And in the case of, and in the heterochromatic context, but this is exactly what I explained to you just right now. The target regulatory sequences are recognized by transcription factor, but that do not activate POL2. They recruit histone modifying enzyme like HAT or methyltransferase, and then you start again with the classical concept. Okay. So this is a summary of all the discussion that we can have. All right. Okay. So just to explain you, because this is quite funny. I mean, it's very, very interesting experiment. So I talked to you about these modifiers and these writers and readers. Okay. And I just want to show you how it was discovered a long time ago, actually. It was because I think it's important that you understand the scientific strategy in order to understand the processes. So these readers and modifier were the first one. They were identified in Drosophila using this. So you know a little bit of genetics of Drosophila. You study it a little bit. Yes, okay. But one very commonly used phenotype is the color of the eyes. So we have the wild type Drosophila, it has red eyes, and we have mutants that have white eyes. It's not white eyes, sorry. It's not important to know. Anyway, this is just the schematic. You don't need to know that. But the gene, there is a single gene that is controlling the color of the eyes. So very simple model. And this gene is carried by one sexual chromosome. It's not a problem, okay? So they use this model. So the idea is quite complicated. So here, it's not complete. It's very, you will see it's very clever. It's very simple and very clever strategy. So the idea was we take a white Drosophila, so white eyes Drosophila. So it means it's a mutant of Drosophila. It has white eyes. Do you know homologous recombination? Have you ever heard about that? We cannot do homologous recombination in plants. We can do that in animals, in yeast as well. Do you know or not? Tell me, because it's quite important to understand the rest of the slides. You know or not? If you don't answer me, it's going to be difficult for me.
I'm not sure if I know.
Okay, so I just write you something on the. Okay, I changed the sharing. 1 second. I explain you this because it is important. Okay. You see the white
of the chromosome.
Yes.
Meiosis?
Yes. Alors, so okay, this is right, but we can use homologous recombination in order to do transgenic animals or to change some genes. So you see the whiteboard? Okay. So for it's homologous recombination. Okay, so the idea is if you have, this is a part of the genome, okay? Here, and so it's a method in order to modify to make transgenics. We use homologous recombination to make transgenic mice, to make transgenic Drosophila, and to make transgenic yeast, but not plants. It doesn't work on plants. The idea is, for example, so this is the genome. It's a part of the genome of the target organism. All right. We have a vector, which is a plasmid. Okay. So that's a vector. And in this vector, we can put a gene of interest. Okay. I will explain you interest. Okay, and on the both side of the gene of interest, we had sequences. So they are quite long. They can be around 200 base pair. We can put more till 1 KB, something like that. Anyway, 200 is more than enough. And these sequences are completely homologous to two sequences that we know in the genome. It means that this sequence in the genome, we know exactly where they are because it's you that you choose them. So once we are in this situation, what we can do, you see the mouse here? Yeah. Okay.
Yeah.
So this is the genome. You want to transfer this gene of interest at this place, not there, there. So you are going to take one sequence here, one sequence there. So from one side and another side of the target locus, and you put those sequences on both sides of the gene of interest, which is contained in the plasmid. You enter this plasmid by transfection into the cells, and what is going to happen? Homologous recombination is going to exchange the strands of DNA. And the result of two recombination will be the genome. You will have the gene of interest, so I call it GI, the gene of interest that has been integrated around the two target sequences. And this is a modified genome. Do you understand the idea? It's okay? So it means that because of the homology between two sequences, we can swap what is in between from the vector to the genomic DNA. So it's a way to transfer genes, and it's the way to transfer them at a specific site. And this is what is important for us. So you understand at least the concept? Okay, so we go back to our flies. So here, what we have, they took a white fly, and using homologous recombination, they reintroduce in that place of a chromosome, I explain you afterward, the wild type red gene. Do you understand the process? So we have a fly that has white eyes, and you reintroduce in this fly, you reintroduce the wild type genes. So the fly has now the eyes are red back again because you complement the mutation. Okay, so I explain you how the drawing, what corresponds to the drawing at the bottom of the picture. In Drosophila, the sexual chromosome, it's a little bit strange because the centromere is not in the middle of the chromosome. It's at one end of the chromosome. It's not a problem. The drawing is done like that. The small line, black line, represent euchromatin, and the big white box represent heterochromatin in this chromosome. Okay, so this is a normal chromosome of the fly, of the Drosophila. We have centromeric region, heterochromatin, so compact chromatin, and the rest of the chromosome is non-compact. Okay, so it's open to transcription. If you take a mutant fly, so white eyes, you introduce the red loci in euchromatin, you reverse the phenotype. Is it okay for you? It means you put back the wild type gene in the mutant context. So if you put the wild type gene, W+, in euchromatin, perfect restoration of the phenotype. But if you choose, because of the use of homologous recombination, you can choose the target of the site of insertion. If you decide to reinsert the wild type gene, W+, but not in euchromatin, but just close to heterochromatin, look what happened. So according to you, why, when it's really in euchromatin, the complementation works, and when it's very close to heterochromatin, complementation does not work? Do you have an idea of that?
Is it because the position near the heterochromatin makes the gene harder to be transcribed? Yeah.
Exactly. You are completely right. Because heterochromatin, what we saw is it means that we don't have gene expression. So they did put it just before heterochromatin, but you see the gene, the wild type gene expression is not good at all. Okay. So the idea, so you understand this experiment. It's very simple. You put wild type gene, euchromatin, you restore the phenotype. You put the wild type gene close to heterochromatin, you don't restore completely the phenotype. Okay. And the researchers were very, very clever to use this phenomenon to understand what are the writers and the readers. So what they did, they took this fly, okay, this one. It's very important. So they took a fly that contained the wild type gene close to heterochromatin. And then they took this fly and they regenerated from this flight. Okay. So they were screening for modifier of the phenotype they were having. So here we have got, so there will be three different drawing one by one to explain you how it works. So here you see back again the structure of the chromosome. So the situation that we just saw, okay? Centromeric region, heterochromatin, euchromatin, and the famous wild type gene that we put just before heterochromatin. The phenotype, we know it, is the one that we saw just before. So you do not restore completely the phenotype. You take this fly and you do mutation. Some mutant, they were affected by in the chromatin condensation, but they didn't know that at that time. So they found flies and mutant of this fly, where they call that a suppressor. Suppressor, why? So keep in mind that the drawing on the left, they didn't, they had only access to the phenotype. So some of the fly that were used for the mutagenesis on the top, when you mutate them, you restore the phenotype. But you restore the phenotype without changing the position of the wild type gene. So it means that between this fly, very weak expression, and the new one, the mutated one, you didn't change the position of the gene, but it is more expressed. So this increased expression was due to the modification of the heterochromatin landscape. So it means that in this fly that has red eyes, actually in the genome of this fly, the wild type W plus loci was not anymore in heterochromatin as it was before. So because the place of W2 in the genome didn't change, it means that it is the heterochromatin place that changed. You understand? Okay. So they found plenty of different, what they call suppressor of PEV effect. PEV effect is position effect variation. So they found suppressor of this effect, and they found activator. So activator is exactly the opposite phenotype. So the original fly, weak expression, because W plus is just at the border between hetero and euchromatin. Suppressor, the W plus is now only in euchromatin, but still at the same place. So strong expression. And the enhancer of this position effect, the wild type gene W plus was not expressed at all anymore. You see, it's very more weak than in the first one. And this is because now the W plus gene, before it was just at the border between hetero and euchromatin, now it's completely in the middle of heterochromatin, so no expression at all. Do you understand this process? It's okay or not?
Yes.
So it is very clever process because you use an artificially generated fly, because it's a transgenic fly, okay? And you reuse this transgenic fly to generate mutant and to find different mutation. So this is the strategy. I wanted to talk to you about that because it's very interesting in terms of logic. And so this was done, look at the date, it was in 98, okay? And later on, the researcher, they isolate the genes that were mutated in this category of suppressors and in the category of enhancers. And guess what they found? They found this. For example, one of the suppressor, they call that suppressor of variegation. Don't pay attention to the name. So one of the gene that they found there, sorry, I come back. One of the gene that they found here, responsible for this phenotype, it was heterochromatin protein 1. The other one gives the same phenotype, histone methyltransferase. The third one, exactly the same phenotype, histone desacetylase. So you see all the mutation were targeted to genes that are controlling the heterochromatin formation. HP1, without HP1, you cannot have heterochromatin because it's the protein that condenses the nucleosome together. Without histone methyltransferase, you cannot have H3 tail that is methylated in lysine number 9. So because if lysine number 9 is not methylated, HP1 cannot interact, so no condensation. Then they found also the histone deacetylase. It makes sense also because this enzyme removes the acetyl group, and you need to have the acetyl group removed from lysine 9 in order to have methyl that can be exchanges with acetyl. So basically, from this very simple genetic screen, so how you start from a gene that is not very well expressed because it's just by heterochromatin, how it can be expressed being at the same position, it is because you have mutated, you have completely knocked down some genes that are directly implicated in the formation of heterochromatin. And we saw them here. The three genes here, I just selected them because this is the one that we saw. HP1, histone methyltransferase, and histone desacetylase. Is it okay? Do you understand the idea of the strategy at least?
I completely understand. I have one question in the previous slide.
This one?
Yes, I'm a little confused about the writing on the slide. Why is it like wild type white gene, even though it supposedly gives the fly red eye?
Okay, so why W? Okay, W doesn't stand for white. Okay.
Yes.
It stands for wild type.
Yes. I'm talking about the writing.
Which writing?
All of them. It has wild type white gene.
No, the wild type fly has red eyes. Sorry. On the left, it's the wild type. On the right, here. On the left here, the white-eyed is the mutant, and the red-eyed is the wild type.
Yes.
It's okay?
Yes.
But do you understand so? Wait, I will try to... One sec. So the wild type, okay. Well, okay. Anyway, no, I'm not going to, because I don't want to write you a mistake. Wild type Drosophila megalogaster. It contains, I don't know where, a gene, we'll call it W plus, and the consequence of this gene is that the phenotype of the eyes is red eye. Okay? W plus give you red eye. Okay? Then what they did, they took the chromosome, this chromosome, that normally does not contain this gene. So, 1 second. So this is the wild type Drosophila here, okay? They used a mutant of Drosophila for this gene. So this Drosophila, the phenotype is white. Okay, because it doesn't carry the wild type red eye phenotype. Okay, is it okay till now?
Yes.
And what they did, they add in a plasmid the W plus gene, so the one that is conferring the color of the eyes to red, and they choose, because you can choose with homologous recombination, they choose either to insert it in euchromatin or just by heterochromatin. When it is inserted here in heterochromatin, in euchromatin, the phenotype is red. And when it's inserted by heterochromatin, the phenotype is red-white, this kind of speckle that we see, just spots. Okay? You understand?
Yes.
So it means that the genetic background at the beginning of the fly, it's a white fly. And they want to complement this mutant phenotype, reintroducing the wild type gene, the red one, but they choose the place where they insert it. Is it better or?
Yes. Okay.
It's okay?
Yes.
Okay, so so to go on, this is how they managed to identify that it was actually protein or enzyme that were responsible for the compaction or decompaction of the chromatin. Okay, so I prepared to you just now 2 slides. So we have almost finished on chromatin. I will just show you a few things in terms of heritability of the epigenetic marks, just to show you what are the laboratory methods that we use to study the chromatin structure, because I think it's important for you to, now you have the background in terms of conceptual data, but technically, how do we study chromatin structure in research in the lab? So there is, it's mainly based on the same strategy. So I'll show you first the strategy, which is called chromatin immunoprecipitation. Have you ever heard about that, or is it completely new for you? Do you know the strategy? It's a general strategy in biochemistry or molecular biology, which is called immunoprecipitation. Do you know the concept, at least?
Yes.
Yes. Yes.
Okay. So the idea is quite simple. If you have antibody specific to one of the target, you can use this antibody to precipitate only the target. Okay, but here it's nothing special. But instead of precipitating some protein, we can precipitate some chromatin state. So the idea is extremely simple. If you are familiar with immunoprecipitation, is you take the starting material is quite important and quite tricky to have. We extract chromatin, and to extract chromatin, it's not the same protocol than to extract DNA, because you all already extracted DNA, I suppose. It's very simple and very fast. But if you want to extract chromatin without really perturbing all the interaction, you first need to isolate nuclei. It's very simple to do. It's a simple centrifugation, and then you can extract the chromatin. So the chromatin, I already told you, it is the complex of DNA plus all the protein that we have linked to DNA. So first step, we extract chromatin. Second step, we used this molecule, which is very widely used in microscopy. It's called formaldehyde. And formaldehyde is a molecule that will create covalent link between all the components that are in interaction. For example, if you have a protein complex that are interacting only with a weak link, so no covalent link, if you treat with formaldehyde, you will find the covalent bridge between the partner. So it's a way when we fix the material with formaldehyde, you block completely all the interaction of all the partner in the complex. So first step, you extract the chromatin. Second step, you cross-link everything using formaldehyde. So the system is blocked from this step. Second step, we randomly cut this chromatin using a physical method. We can use sonication. Sonication will break DNA randomly. So from the full chromatin linked, cross-linked with formaldehyde, we have now plenty of different fragments. And the way the drawing is made, so you see this little piece of red DNA, there is a protein that is interacting with it. So if we follow the drawing, not all the fragment will carry the protein, but if we have an antibody specific to the protein, we can separate the fragment containing the protein from the fragment that do not contain the protein. And at the end of this, it's a simple immunoprecipitation, but we precipitate chromatin, that's all. And at the end, what we can do is collecting all the purified complexes, and these complexes, they are DNA plus the protein, and we can sequence, clone them, and sequence them. So chromatin immunoprecipitation, it's a simple procedure, it's a simple immunoprecipitation, but it's very powerful because of this step. We have plenty of antibody extremely specific against different histone with different post-translational modification. In other words, you can buy, there are companies that sell that, you can buy an antibody that is able to recognize specifically, for example, histone H3 that are methylated at position 9. Okay? Now, from that, you understand if you want to have the global view of all the fragments of the genome that are directly in interaction with H3K9 methylated, all the fragments that you will be sequenced, they will be in that situation. Do you understand the process like that? Yeah, it's okay. So it's a classical immunoprecipitation, but the target are modified, specifically modified histone profile. So this is a very old strategy. It works very well, but it's not high throughput. So nowadays, we scale up this immunoprecipitation. The strategy is exactly the same. We still use specific antibody, but it's just genome wide and really much faster. So this is what I show you in the next slide. And we call that chip on chip. Alors, it means okay. It means chromatin immunopurification, so chip on chip, because chip are microarray DNA chips. And if you look at the procedure, it's exactly the same, except that at the end, we don't clone and we don't sequence the product that we purified. We use directly the result of the experiment in a microarray to have all the genes at the same time. So the strategy is like that. Same first steps, cross-link and sonicate, exactly the same as before, as the slide before. So you see the antibodies that can recognize the histones. So you immunoprecipitate with specific antibody. There are two categories, what has been precipitated and what has not been precipitated, or the input. You manage to label the antibody with different fluorescent dyes, and then after all these products, so with different fluorochrome, you hybridize them on genomic array. And there are software after. So you know about genomic arrays out there? Do you have, just to know if you have an idea?
The microarray, the DNA.
So there, yeah, so you have the microarray, you know. I think that you know the one that you know the most are microarray used to quantify gene expression. Is it?
Yes.
Okay. So on microarray where you quantify gene expression, all the spots on the array, they correspond to cDNA, okay, to all the genes. But we have genomic array. Genomic array is the same strategy, same technique, but instead of having only the cDNA, it has all the fragments that cover the whole genome. It means coding and non-coding. So the whole genome on an array. And we know the position of every spot. So when you use this strategy, it is exactly immunoprecipitation as we saw before, exactly the same, but at the end, instead of sequencing, we use an array. So from this drawing, I hope you realize that all the fragments that have a red fluorescent dye linked, it is because they contain, they were purified by the antibody. So all these fragments of DNA, if we hybridize them on the array, you will see on the array a signal only on the part of the genome that contains this protein. So you can have a direct picture of the whole genome that contain, and the places in the genome that contain the target of the antibody. You see the difference between this slide. The technology is the same. Eh? This slide, we use antibody, but at the end we need to sequence. And this one is exactly the same. Instead of sequencing, we hybridize that to a full genome microarray in order to know which part of the genome is in that situation or another situation. Is it okay for you? So this is what we call chip on chip. It's very powerful because it's like if you take a picture of the state of the chromatin at the whole genome level. And obviously, you can use different antibody. For example, you can use antibody directed against H3K9 acetylated, but you can have also antibody against H3K9 methylated, and so on and so on and so on. So with this strategy, we can have different picture of the genome for different post-translational modification of the histone at the same time. Okay. Is it clear for you? Yes or no? Okay. Okay, so to finish, because we are a little bit late, so I think we need 20 minutes maximum, let's say. Actually, you are not late, but I will be late. So just we won't, in this teaching unit, BP10, we won't spend a lot of time on that because it's a very complicated work, but I need to explain you. So in this slide, it's a summary of what we saw concerning chromatin conformation and transcriptional activity. And I just add one part, which is in red here, that concerns DNA methylation. Okay, so just a quick summary. We know that all what is in orange, we already talked about that yesterday and today. So two different states of chromatin, open or closed, transcriptionally active or transcriptionally inactive, and the histone modification that we saw. Acetylation, desacetylation, methylation, and so on. Okay, so this is what we saw. And there, for quite a long time, do you know that DNA, the genomic DNA, can be methylated in some places? Have you ever heard about that? Do you know or not? Tell me.
Can you repeat? I didn't get that.
Do you know? Have you ever heard about the methylation of DNA of the site?
Yes.
Yes.
Yes. Okay. So this is another very important. So I will make a very short summary on that, just to show you something, because so. 1 second, because it's better that. So we saw from the beginning when we talk about chromatin structure. So we've got two states: condensed and non-condensed. Okay, that's very good. So it's controlling the transcription. When it's open, it's okay. When it's closed, it doesn't work. So this is for one cell. But what is happening when the cell is dividing? When the cell is dividing, you understand that the two daughter cells need to have the same territories of heterochromatin and the same territories of euchromatin. Is it okay? So the compaction of chromatin need to be heritated. Is it okay that? Okay. So researchers searched for a long time on what was the mechanism in order to transmit the state of compaction of one cell to the daughter cells. Because the problem is that when a cell enters mitosis, all the DNA is decompact. So after mitosis, the DNA needs to be recompacted, but with the same pattern. So the question was, what was guiding the same pattern of compaction of chromatin? You understand the question? Okay. So first finding is that, and this is what is written in this table, all the time when we check for heterochromatin, and if you analyze the sequence of DNA, okay, which is in heterochromatin, we found that most of the time the DNA at the cytosine, because this is only cytosine that can be methylated, the DNA is extremely methylated. But on the other side, all the regions in a cell, most of them that are transcriptionally active, which means euchromatin, the DNA in this chromatin is relatively unmethylated. So technically, they found a correlation between the compaction of chromatin and the level of methylation of DNA. Do you understand this idea? It means that all the DNA which is inside an heterochromatin island is going to be mainly methylated compared to euchromatin. So cytosine methylation is always associated with heterochromatin formation. And from that, so I make a very, very quick summary. So there are only 2 slides on that. I show you what is the most relevant model because it's not yet very clear, but this one is really working. The model which implies DNA methylation, okay, at the DNA level, and histone post-translational modification in order to be able to have the compaction state that can be inherited from one mother cell to the daughter cells. So there are 2 steps here. There are 2 different, okay. So 2 concepts, and I'm going to explain you. This is the 2 concepts, only 2 concepts that has to be remembered. The DNA methylation, so methylation of cytosine, serve as a guide to de novo histone post-translational modification. What does it mean? It means that on a piece of DNA, of genomic DNA, if the cytosine are methylated, they will be recognized by complexes, so writer complexes. If we keep the concept of writer and reader, they will be recognized by writer complexes that are going to modify the histone post-translational modification, and this modification will give rise to compaction. So, on the other hand, the methylation, so you take a piece of chromatin, which is heterochromatin, and the DNA is not methylated. If this piece of chromatin contains histone that are methylated, like H3K9 methylated, so it means heterochromatin this. The consequence of having the histone in a state that is compatible with compaction has a consequence, which is the methylation of the DNA that is inside this region. Do you understand what I say? Because it's quite tricky. So if the DNA at one place is methylated, the consequence will be the methylation of the histones. And if the histones are methylated in one place, the consequence will be the methylation of the DNA. So it's reverse. You understand the concept? Okay, and here we have a partial explanation of the phenomenon. So the schematic, you have to read it from left to right. So left, it's the DNA of the mother cell, and right, it's the duplication of DNA. So it's written here, a replication of DNA methylation pattern. So here we are, so you've got the legend here. Dark blue, this is old histone, and new light blue will be the new histone. It makes sense because you double the DNA content, so you need to double the amount of histone content. Okay, so that's fine. So there are the old histone and the new histones. Then the little red bars, spotted bars, they mean covalent modification. So covalent modification on histones. Okay, so this is, for example, the methylation of histone 3K9. Okay, then the little black stars, they show you when DNA is methylated. Here in red, we've got a protein which is able to bind to methylated DNA, methylated cytosine. And in green, we've got enzymes that are able to modify the histones. So, for example, one of this histone modifying enzyme can be histone acetyltransferase, histone methyltransferase. It's a writer. Okay, it's not a reader, it's a writer. Okay, so before cell division, the state is like that. We are in a situation of heterochromatin. We have covalent modification on the histone. Let's say that it is a methyl like H3K9 methyl. Okay, so chromatin is compacted, and the consequence of compaction of chromatin is DNA in this region is methylated, more methylated than other places. Then, after duplication, after replication, you have only one strand of DNA which is going to be methylated, and the other strand won't be methylated. Okay, it's semi-conservative replication. So we have, at that stage, one strand of DNA methylated, the other strand not methylated. And for the other double-strand DNA, it's exactly the same situation. One strand methylated and the other one not. Okay. From that, we have, okay, so it's subsequent. So the histone that were in a conformation of compaction, they will be redeposited on both strands, but you have new histone that need to form new nucleosome. So the one that were already modified, methylated, will stay methylated. It's not a problem. But what's happening is, as soon as we are in this context, so you see that the new two double-strand DNA are not completely compact because part of the nucleosome are methylated and part are not. So it's not a convenient situation. And what is happening is you have specific proteins that are able to recognize the methylation of DNA. And the methylation of DNA, once this protein is recognized the methylated DNA, is going to interact with an histone modifying enzyme, like an histone methyltransferase, for example, in order to, this is the two small dark arrow, to remodify the histone post-translational modification. Do you understand the process in this drawing? So it means that this semi-conservative event is very quickly rebalanced in order to be completely in a condensed situation. So there is another way to see it. I put this drawing because it's a little bit more clear, maybe. So here the color code is, so in yellow you've got the DNA with the two loops around the histone core. The small balls here, which are light purple, are the cytosine on the DNA. So when it's light purple, they are non-methylated, and when they are dark purple, it means methylated cytosine. And so in this way, you can see what's happening here. So when we've got the H3 tail that is methylated, okay, this situation, we have this HP1 that is recognizing the methylated histone. So this is the protein that is responsible for condensation of chromatin. But you see that non-methylated, as soon as a cytosine was methylated, okay, then you have complexes that are formed and you have this new partner. So this is the only one to remember, maybe. And the name is DNMT3. And it means de novo methyltransferase. So it's a DNA methylase that will methylate de novo DNA that is not methylated when the nucleosome are harboring a methylation of the H3 histone. Because methylation of the histone tail, H3, means heterochromatin. And as soon as the DNA is inside heterochromatin, it needs to be methylated in order, the aim is that the information is going to pass to the daughter cells. And the next step is what we saw here. So the two daughter cells, they are carrying half of the modification, and you've got all the system of recognition that is going to be able to reset the system in the same state as the one that you find in the mother cell. Is it more or less clear what I wanted to show you? So it's very simple. We've got cells with patches of heterochromatin, euchromatin everywhere in the genome. Under every region which are in the heterochromatic state, the DNA is methylated. The methylation is partly transferred to the daughter cell after mitosis. This methylation is then directly compensed to be fully methylated. This is what we see here. So now we have both strands that are methylated. And as soon as both strands of the new cells are methylated, there are enzymes that are modifying the histone that are recruited in order to be back in the same state. Okay? Okay, this is a summary of what I just tell you now. You have to understand that it is a self-reinforcing repressive cycle. Do you understand? Because DNA methylation. So here you've got all the partners. So take care, because you tell me if you see very clear. Here we've got the nucleosome. When it's written K9, it's the lysine number 9 of histone H3. So in this drawing, you see that it can be acetylated. Acetylated means non-compact. It can be methylated. Methylated means compaction. Compaction, when it's methylated, it interacts with HP1, and HP1 interact each other, so there is condensation. And look here, if you see the spot of the mouse, there is another methyl in green. It's not very well done, but it is the methylation of DNA. Okay. And look, what's the concept is that if you have methylated H3 on lysine 9, there is interaction between HP1, and interaction between two different HP1 give rise to compaction, but HP1 interact with this red enzyme, which is a methyl transferase of DNA. So it means that when we are in a configuration of heterochromatin, there are enzymes that are going to methylate the DNA in order for the next cycle to re-put the same mark on the histones. Okay, so in this drawing, it's quite complete, because you see that also when lysine 9 are methylated, it means heterochromatin. We have HP1 interacting, but also it recruits histone desacetylase. And why it recruits histone desacetylase? Because you need to have the activity of removing an acetyl group on this lysine in order to put new methyl. Here it's histone methyl transferase. So do you understand the global meaning of this drawing? Because it's very complex. There are a lot of data there. Okay? So heterochromatin stimulate histone methylation, stimulate DNA methylation, inhibits desacetylation, that will remove the acetyl, so stimulate desacetylation. And this is why we call that a self-reinforcing cycle. All right. Do you have any question? We're almost finished now. Okay. So just tell me if you have any question.
Just can I ask, because in the replication, so the CpG of DNA is will remain to the in the daughter strand, right? So the protein quicker give the methyl group to the histone will depend on that CPG, the state of CPG isolate to give the.
Yes, it's there. I mean, you're talking about the methyl transfer to histone or to DNA?
Can you go back to the previous slides? I think
This one.
it's more clear.
Yeah.
In the daughter strand, I can see that in the I can see the CPG state of the DNA it remain in the new strand, right? After replication, the CPG of the DNA it will be remain in it.
Here you mean?
Yeah.
Okay. I have not done this. No. Okay. You can imagine that here you have also another methyl CPG binding protein here, just here, and it will recruit also an histone modifying activity here. Is it that problem or?
No, I mean, if I want to know which will go first, like the DNA CPG methylation will go first, then the histone methylation will go after in the new strand.
Okay.
Yeah, that can.
I cannot answer that. I cannot. I never saw any clear. I would say, to be sincere, I don't know if we know. I think there are. If you see the model, there should be two processes that happen at the same time, because histone methylation. Look here, histone methylation has for a consequence to recruit DNA methylase, okay, and methylated DNA recruit enzymes that are modifying the histone. So it recruits also like histone desacetylase or histone methyltransferase. So to be sincere, it's like the story of the eggs and the chicken. I don't know where to start, but this is why and the problem all the time when you have this kind of cycle, which is self-reinforcing, it's very difficult to say what is the first event that starts the cycle. To be sincere, I don't know if it is the copy of the methylation of DNA that occurs first or the methylation of the new histone that occurs first. Okay. But what you have to keep in mind, which is more fundamental, is that there is a direct biochemical link between the state of methylation of histone and the state of methylation of DNA. And the state of methylation of DNA, there is only thing that is very clear, is that one strand of the DNA of the mother cell is going to be so methylated at some regions, is going to be found back in one strand of the DNA of the daughter cell. So it means that here we already have a template to put a second methylation here. Then it's true that we have already nucleosomes that are in a condensed situation. So I would say that it's more the duplication of methylation of DNA that should occur first, but almost at the same time, we've got all the nucleosomes that are going to be methylated as well. Okay. You see what I mean? It's very difficult to tell you who starts first.
It's more clear now.
Okay. Yeah. So just, did you have other question or is it okay? Okay. So just to finish, I just show you one result. It was the very first one. It was done in plants. It has been published in Cell, which is, I think, one of the best paper in biology. In 2006, and what they did the first genome-wide high-resolution map of DNA methylation in Arabidopsis. And this is very interesting. I just show you one figure of that. So it's a very complex figure. Wait, I'm going to, well, okay. So we're not going to look at the top of the figure because it doesn't make sense for this lecture. The top part of the figure is all the methodology that they use to quantify it. And I want that we focus on the red box here. That is very easy to understand. So I explain you how to read it. So this is the full-length chromosome number 4 in Arabidopsis thaliana. Okay, so from the north of the chromosome, position 0 to the bottom. It's something like 15 or 16 megabase. The little white circle is the centromere. So you see there are gray boxes and dark gray boxes. So this is to show you where the heterochromatin is present in this chromosome of Arabidopsis thaliana. So we always have heterochromatin in pericentromeric regions. Okay, there are plenty of repetition there. So the centromere is here. Pericentromeric regions, they are completely condensed and heterochromatin. And one particularity of this chromosome number 4 in Arabidopsis is that in the north arm of the chromosome, there is, we call that an heterochromatic knob. It's a loci, a locus here, that contain a lot of repetitive element, and therefore it's completely compacted at that place. So the idea is you take this chromosome, full-length chromosome, we know already which are the parts that are heterochromatic and which are the part of the chromosome which are euchromatic. What they did, so it's a little bit difficult to understand. So here down there, you've got 3 boxes. Okay, these 3 boxes, we're not going to look at the bottom part of the boxes because we have to read the full paper to understand, and it's not the aim today. We're going to see only the 3-4 lines. So the little line with the blue data, the green one, and the second group. So what they did in these 3 boxes, left, middle, and right, you have the result of, for example, this box on the right correspond to the analysis data on the very top of the north of the chromosome in euchromatin. Okay, this is a place where chromatin is not condensed. The box in the middle correspond, you see the little arrow here, correspond to a place in the chromosome that is very close to the centromere and right in the middle of heterochromatin. So it's data typical for heterochromatin. And the box on the right corresponds to another place on the south of the chromosome, which is also like the first one, euchromatin. Okay, so as a summary, this box is euchromatin, heterochromatin, and euchromatin. And here on the first line, so we're going to look at only the 3 lines here. In blue, they show you in this fragment. So you have the size here of the scale. So imagine that this window is going to be 30 kb in that region. This window here is going to be 30 kb in that region here. And this one is a smaller one. You see it's almost, it's going to be 5 to 6 kb in that place here. So for all this fragment, they report on the very top line the presence of repeats of DNA. Okay, so what can we see? We can see that the region on the north of the fragment does not contain or very few repeated sequence. Okay, so very few repeated sequence. The sequence on the south of the chromosome, which is also euchromatin, also contain a small part of repeated sequence and after no repetition at all. Whereas the part of the genome that is located just by the centromeric region contains, it's mainly only this, contain a lot of repetitive elements. So that's the starting point. And the two lines just below, the green one, the blue graph here, they are giving you in wild type plants, so no mutation. This is why I don't want that we look the bottom, because they are mutant. So in wild type plant, they check the level of methylation of DNA. Okay, this is the title of the paper. And why there are two lines? Because they use two different methods. The first method to analyze the methylation of DNA is methylcytosine immunoprecipitation. So you remember chromatin immunoprecipitation. We were using antibody directed against one particular form of histone in order to know where they were in the genome. But we can do the same for methylcytosine. It means that we have, it is commercially available, antibodies that are specific to cytosine when they are methylated. So it means that with this method, we can separate the pieces of DNA that are highly methylated compared to the ones that are not methylated. And so this is the result obtained with this method. And the second line, the blue one, it's the same data, but that have been obtained with a different method that use methyl DNA binding protein. It's not important to remember. It's another method to analyze the methylation profile in the DNA. So first, you can see that the green data, they are the same as the blue data. Okay, so we can only see the data. So what can you? We will finish with this slide. How can you interpret these two graphs? Can you give me at least one idea? Do you find a rationale between what I'm showing you now and all what we saw before in the lecture? Do you have an idea?
I can see here is the highly methylated.
Can you speak a little bit louder, please?
It's highly methylated
Oh,
because the signal was quite dense, quite high.
yes, but it's highly the DNA is highly methylated, but not everywhere.
Yes,
So
but it's related to the repeat in the heterochromatin.
exactly. So what we found is when we are analyzing DNA that is in heterochromatin, we found that first, heterochromatin is there to protect for repeated elements. Okay, and this DNA is extremely methylated. So the fact that this DNA is very methylated, it means that because the DNA is methylated, it will recruit histone modifying enzyme that is going to make the histone and then keep the chromatin compacted. Okay? So it makes sense. On the other hand, if we take the two regions that are supposed to be in euchromatin, we don't see very much methylation. But if we look at this part, the south, we can see that there is no methylation. Don't pay attention to this white arrow because it was specific for the paper. So you see that most of the region is not methylated. It makes sense because this is euchromatin. But look, there is something that is very interesting here, is that in this euchromatic region, there is a place where you have some DNA repeat. It can be transposons or it can be microsatellite or whatever, but a very small stretch of repetition. And as soon as the cells detect repetition, the DNA will be methylated, and therefore it means that The drawing on the top is not perfect, but there is a small place where this portion of the genome is under heterochromatic situation to block the repeated sequences. Okay? And so you see there is a clear link between DNA repetition, DNA methylation, and the consequence of DNA methylation will be heterochromatic formation at that place in order to block the mobile element. Okay? And you see clearly that also in this part, for example, here, we are 100% heterochromatic, but there are places where there are no repeats, even if it's mainly heterochromatic. And those places where you don't have any repetition, DNA that is in those loci, it's not methylated. On the other hand, you have places, and that's the case, this is why they show you this example, places in the genome, for example, the north of the chromosome, of this chromosome, we don't have repetition, except at the end there are repetition, where you see methylation of DNA, perfect, like there. But you have some places where the DNA is locally methylated without any heterochromatin information and moreover without any DNA that is repeated there. So it's another more complex process. It's okay. So this was an example to show you the link between DNA methylation and histone modification and compaction of DNA in the context of heritability of the marks. Okay, we finished now. We will go on. So one thing. So 2 points. So for now, we covered during these 3 days all the structure of the genome and all the structure of chromatin. And what we saw in chromatin, it's the link of the chromatin that can be in 2 different states. And we already talked about the importance of these 2 states in terms of transcriptional regulation. For now, we won't talk a lot about this kind of things, but we are going to study really in depth the mechanism of transcriptional regulation. And you will see, we'll come back a little bit sometimes on histone modification. But for now, we can consider that all the chromatin structure, we already almost covered everything. So just also a little bit an update. So tomorrow we won't have the lecture as planned because I was informed yesterday that I have to attend an important meeting for the PhD grant at USTH tomorrow morning for me. So I am really confused, but I cannot be at 2 places at the same times. And it's a question of funding for the students for the PhD grant. So tomorrow we don't have the lecture tomorrow, and Friday morning for me, afternoon for you. You can check the timetables. Miss Hang did the update, so we will start for you at 2 p.m. Vietnam time. So it will be 8 o'clock for me, French time. And we will spend the 4 hours if it's okay for you. Is it okay? You tell me. So no lecture tomorrow and Friday, 2 p.m. And then maybe you can tell me now, I send Hang this morning the PDF files for all the slides that we saw. Did you receive them?
Yeah, but we need password.
Yes. Okay. This is so the password is very, very complicated. You type so in caps. You type BP10 2022.